Welcome to the podcast. Today, we're exploring the fascinating world of ancient epidemics and how modern bioinformatics is rewriting history. Scientists are now using advanced DNA sequencing and computational analysis to understand how diseases emerged, spread, and evolved, and how our ancestors fought back. The key to this is ancient DNA, or ADNA, recovered from archaeological sites like human bones and teeth. Previously, studying old diseases meant looking for visible marks on skeletons, which missed many fast-acting infections. But now, next-generation sequencing allows us to reconstruct the genomes of ancient pathogens, like bacteria and viruses, giving us a molecular fossil record. So, what have we learned? We can now pinpoint the exact germs behind historical pandemics. For instance, Yersinia pestis genomes from Black Death victims confirmed its role and helped trace its evolution through various plague outbreaks. We've also identified ancient tuberculosis, leprosy, smallpox, and hepatitis B. By comparing old pathogen genomes to modern ones, we can track their evolution, seeing how they became more dangerous or transmissible. Studies show that major plagues often came from new strains of Yersinia pestis*, with its earliest human infections dating back to the Bronze Age, long before big pandemics. We've even found extinct pathogen versions, giving us a fuller picture of past disease landscapes. This research also reveals how diseases jumped from animals to humans. For example, an ancient Yersinia pestis genome was found in a 5,000-year-old sheep, directly linking animal reservoirs to human infections. We're also learning how human migration and lifestyle changes, like the shift to farming, affected disease spread. Denser populations in agricultural settings likely saw an increase in certain pathogens. These ancient genomes also help us accurately date when pathogens emerged or gained new traits. Our ancestors weren't passive victims. Infectious diseases were a huge evolutionary pressure. By studying ancient and modern human DNA, scientists can find genetic traits that became common because they helped people resist specific diseases, especially genes tied to our immune system. This gives us a direct look at how humans and microbes co-evolved. Interestingly, some genetic traits we inherited from Neanderthals and Denisovans may have helped modern humans fight new pathogens as they moved out of Africa. However, some past adaptations might now, in our modern world, make us more prone to inflammatory or autoimmune conditions. How is this all done? It involves careful DNA extraction from ancient samples, often using techniques to enrich tiny amounts of pathogen DNA. Then, high-throughput sequencing generates massive data, which sophisticated bioinformatics tools analyze to piece together genomes, confirm their ancient origin, and identify specific pathogens. But it's challenging. DNA doesn't always preserve well. Contamination with modern DNA is a big risk, and ancient DNA is often very fragmented. Plus, telling a true pathogen apart from a harmless microbe can be tricky. So why does studying ancient epidemics matter today? By understanding how pathogens evolved, how humans adapted, and what drove past pandemics, we gain crucial insights for tackling current and future infectious disease threats. It offers a long-term evolutionary view for modern public health, helping us prepare for emerging diseases. This work isn't just about the past, it's about safeguarding our future. Thanks for tuning in.